Take your shoes off. Right now if you can. Set them beside you and press the soles of your feet flat against whatever surface is beneath you. Feel the temperature differential. Feel the texture registering through the skin. What you are experiencing in this moment is not merely comfort or sensation. It is the activation of a sensory reporting system so ancient, so deeply wired into your central nervous system, that the human brain spent approximately 3 million years building it before a single piece of footwear existed on Earth. The shoe is, in geological terms, brand new. The system underneath it is not. And when you remove that shoe and place bare skin on bare earth, you are not doing something alternative or therapeutic or ritualistic. You are restoring a sensory input channel that your brain's architecture was literally built around, and whose absence the evidence increasingly suggests may be doing things to your neurology that we are only beginning to measure. The foot is one of the most densely innervated surfaces of the human body. The plantar surface, the sole, contains approximately 200,000 nerve endings per square centimeter in the highest density zones near the ball and heel. These are not simple pressure receptors. They are mechanoreceptors of at least 4 distinct types. Meissner's corpuscles, which fire on initial skin deformation and detect light touch and texture changes. Merkel's discs, which sustain firing under constant pressure and encode fine spatial detail. Ruffini endings, which are exquisitely sensitive to skin stretch. And Pacinian corpuscles, which respond to vibration in the frequency range of 40 to 400 Hz. Each of these receptor populations sends its signal up a different fiber class, processed at different speeds, converging in the somatosensory cortex in a dedicated region that, in the famous cortical homunculus drawn by Wilder Penfield at McGill University in 1950, is disproportionately large. The foot takes up far more cortical real estate than its physical size would suggest. Your brain has always treated the foot as an important organ. We treated it as something to cover. When you walk barefoot on an irregular surface, earth, grass, gravel, sand, something specific happens that does not happen on a flat, cushioned surface. The terrain variability forces constant microadjustments from the ankle, foot, and lower leg musculature. This is obvious and mechanical. Less obvious is what it demands from the nervous system above the ankle. The cerebellum, which coordinates balance, proprioception, and fine motor timing, receives a continuous high-bandwidth stream of novel input that it is required to process and respond to in real time. Research published in 2017 by Daniel Lieberman's lab at Harvard University, analyzing barefoot gait mechanics across multiple populations including Kenyan runners and adult Americans, established that barefoot walkers automatically shift toward a forefoot or midfoot strike pattern. The heel impact that defines shoe-cushioned walking essentially disappears when the heel is unprotected. This is not a conscious choice. It is reflexive load management. The nervous system detects the absence of cushioning and reorganizes the entire kinematic chain from the ground up. The proprioceptive signal from the sole changes the motor program in the brain. The foot talks to the brain and the brain immediately listens. But there is a second mechanism operating at the same time, and it runs through completely different circuitry. The skin of the human foot, like all skin, contains interoceptive and exteroceptive pathways that connect to the autonomic nervous system, the branch of your nervous system that runs heart rate, digestion, immune response, and the balance between sympathetic arousal and parasympathetic rest. Irregular natural surfaces activate what researchers call low-threshold mechanoreceptors, receptors that fire under gentle, non-painful contact, and these project to lamina I of the dorsal horn of the spinal cord, a gateway region that has direct ascending connections to the insular cortex and anterior cingulate cortex. Both structures are central nodes in the brain's interoceptive network, the system that monitors and regulates your internal body state. D. Bud Craig at the Barrow Neurological Institute in Phoenix spent decades mapping these pathways and published a landmark synthesis in Nature Reviews Neuroscience in 2002 establishing that the lamina I pathway carries not just pain and temperature but the full range of homeostatic afferent signals, including the gentle, sustained tactile input that comes from contact with textured natural surfaces. When the sole of your foot contacts earth, you are feeding input into the same neural network that tells your brain whether your heart rate is too high, whether your body temperature is too hot, whether your gut is inflamed. The signal arrives in the same processing center that generates the subjective sense of how your body feels in this moment. This is not metaphor. It is the anatomical routing of sensory fibers established over decades of work in neuroanatomy. The implications of that routing are what make the grounding research simultaneously compelling and contested. In 2012, James Oshman, a biophysicist with a doctorate from the University of Pittsburgh, and Gaetan Chevalier at the University of California, San Diego published a review in the Journal of Environmental and Public Health arguing that direct electrical contact between the human body and the Earth's surface, specifically, the transfer of free electrons from the ground into the body, reduces physiological markers of inflammation. The Earth carries a net negative electrical charge. Its surface, maintained by the global atmospheric electrical circuit driven by continuous lightning activity around the planet, holds a reservoir of free electrons at approximately -400 millivolts relative to the ionosphere. The human body, when isolated from the Earth's surface by rubber or synthetic sols, slowly accumulates a positive electrical charge. This is measurable. Chevalier's 2013 study, published in the Journal of Alternative and Complementary Medicine, measured surface electromyography and electroencephalography in subjects who sat with feet grounded on a conductive pad connected to Earth versus subjects with feet on an insulating mat. The grounded subjects showed measurable changes in electrical potential across the skin within minutes. The brain activity changes were detectable on EEG. This is where the evidence requires careful calibration, because the story splits into two very different levels of certainty here. The physics is not disputed. Free electrons do transfer across skin contact with the Earth's surface. The body does carry electrical charge. The Earth's surface charge is real and measurable. What remains contested is the downstream magnitude of the biological effect. Chevalier and Oshman's work has been criticized for small sample sizes, inadequate blinding in some studies, and reliance on self-reported outcomes. The 2012 review and several subsequent papers in the same vein have not been replicated by independent large-scale randomized controlled trials. The electron transfer is real. Whether that transfer produces the anti-inflammatory effect at the cellular level, the proposed mechanism involves free electrons neutralizing reactive oxygen species and thereby reducing oxidative stress, has plausible biochemistry behind it, but the direct human evidence is preliminary. The connection is mechanically coherent. It is not yet proven at the clinical standard. What is harder to dismiss is the psychophysiological data. Mark Berman at the University of Chicago has spent more than a decade studying what contact with natural environments does to directed attention and cognitive fatigue. His 2008 paper in Psychological Science with John Jonita and Stephen Kaplan, the paper that introduced attention restoration theory to quantitative neuroscience, showed that a 50-minute walk in a natural setting produced a 20% improvement in working memory performance compared to a 50-minute walk in an urban environment. The participants walked the same distance. The physical exertion was equivalent. The difference was the sensory environment, specifically, the softly fascinating, involuntary attention-engaging nature of natural terrain. Natural environments, Kaplan's theory proposes, allow directed attention networks to recover by occupying the brain with low demand but continuously novel processing. Irregular ground, ambient light through vegetation, unpatterned sound, all of this feeds the brain's bottom-up sensory system without depleting the top-down effortful attention system. The ground underfoot is part of this sensory ecology. It is not incidental. Now I want you to think about the ground itself. Soil, not concrete, not asphalt, not synthetic turf but actual earth, is a living medium. 1 gram of fertile topsoil contains between 100 million and 1 billion bacteria from thousands of species. It contains fungi, archaea, nematodes, protozoa, and a chemical signaling network between these organisms so complex that the Max Planck Institute for Chemical Ecology has dedicated an entire research division to decoding it. Among the chemicals present in healthy soil is Mycobacterium vaccae, a non-pathogenic soil bacterium whose interaction with the human immune system was studied by Graham Rook at University College London and, in the context of mental health, by Chris Lowry at the University of Colorado Boulder. Lowry's 2007 study, published in Neuroscience, found that injecting Mycobacterium vaccae into mice, and subsequently identifying that the bacterium activates a specific subset of serotonin-producing neurons in the dorsal raphe nucleus, produced behavioral changes consistent with anxiolytic and antidepressant effects. The bacterium activated the same neural circuit that selective serotonin reuptake inhibitors, SSRIs, act on. Not through chemical simulation but through immune signaling that ultimately reached a population of neurons in the brainstem that regulate mood, arousal, and stress response. The bacterium interacts with the immune system through pattern recognition receptors called toll-like receptors, triggering a cascade that eventually increases serotonin turnover in the prefrontal cortex. This is the mechanism of contact with soil, not symbolic, not traditional, not cultural. Molecular. The route by which vaccae enters the human body during barefoot walking is not primarily through the skin. Lowry's subsequent work established that inhalation of soil aerosols and incidental ingestion, particularly in children, but measurably in adults as well, are the primary exposure pathways. The skin contact is not the delivery mechanism for the bacterium. But the act of walking barefoot on earth brings you into proximity with soil aerosols in a way that shoe-wearing walking does not, and it brings you into the complete sensory environment of which the bacterium is one invisible component. The direct skin contact with living soil, the aerosol inhalation, the mechanosensory activation, the irregular terrain processing, they are not separate interventions. They are one experience that happens to operate through multiple independent biological mechanisms simultaneously. And here is where the story changes. The hippocampus, the seahorse-shaped structure in the medial temporal lobe that plays the central role in forming new memories and in spatial navigation, is one of the only regions in the adult human brain that continues to produce new neurons. This process, neurogenesis, was for decades considered impossible in the adult brain until Joseph Altman's initial observations in the 1960s, eventually confirmed and extended by Elizabeth Gould at Princeton and Fred Gage at the Salk Institute through the 1990s. The hippocampus generates new neurons in the dentate gyrus, and the survival of those new neurons depends critically on environmental enrichment, physical activity, and stress reduction, specifically reduced cortisol. Kirk Erickson at the University of Pittsburgh tracked 120 sedentary older adults, average age 67, in a randomized controlled trial published in the Proceedings of the National Academy of Sciences in 2011. Half walked for 40 minutes 3 times per week. Half did stretching. After 1 year, the walkers showed a 2% increase in hippocampal volume, a region that typically shrinks by 1 to 2% per year in older adults. The walkers had reversed more than a year of structural brain aging. This is walking as a neurological intervention, not metaphor. The mechanism is BDNF, brain-derived neurotrophic factor, a protein that Erickson's group measured in blood samples and found elevated in the walking group, which promotes the survival and differentiation of new neurons in the hippocampus through activation of the TRKB receptor on the target cells. Now the question is, what happens when the walking occurs on natural irregular terrain, barefoot? The honest answer is that the comparative study, randomized controlled trial, barefoot on dirt versus shod on asphalt, measuring hippocampal neurogenesis and BDNF longitudinally, has not been done. What exists is a convergence of mechanisms that each individually have evidence behind them, and which combine during barefoot walking on earth in a way that no one study has yet captured in full. The cerebellar and somatosensory activation from irregular terrain is real and measurable. The autonomic modulation from low-threshold mechanoreceptor input is anatomically established. The BDNF and neurogenesis pathway from walking is documented in multiple independent trials. The Mycobacterium vacui pathway from soil exposure is biologically coherent with preliminary animal evidence and limited human observational data. The attention restoration from natural environments is replicated across multiple independent labs. These mechanisms do not compete. They operate in parallel. They share some downstream targets: cortisol reduction, serotonin availability, hippocampal BDNF, through completely independent upstream routes. That convergence at shared targets is why the subjective experience of walking barefoot on earth feels disproportionately significant compared to the apparent simplicity of the act. It is not one thing happening. It is 5 things happening at the same time, several of which are pointing at the same brain structures. Here is the part of this story I find poignant. The oldest known sandals, flat pads of plant fiber tied to the foot, date to approximately 7,000 to 8,000 BCE, found in the Fort Rock Cave in Oregon by Luther Cressman in 1938 and radiocarbon dated by the University of Oregon. But fully enclosed shoes as foot protection rather than simple soles did not become widespread across human populations until much later, and in most of the world, barefoot contact with earth remained the norm for the vast majority of waking hours throughout most of human history. The rubber sole, the material that most effectively insulates the body from the Earth's electrical surface, was not mass-produced until the mid-19th century, when Charles Goodyear's vulcanization process made it commercially viable. The concrete and asphalt surfaces that now cover the floors of cities, the surfaces on which urban humans spend most of their walking hours, did not exist at scale before the 20th century. In less than 200 years, humans eliminated 2 inputs that had been present in essentially every waking hour of essentially every human life for 3 million years. Bare skin and electrical contact with the Earth's surface, and mechanoreceptor input from irregular, organic, living terrain. The brain expecting those inputs, because it spent 3 million years being built around them, is still expecting them. It did not get the update. This is not an argument for primitivism or a rejection of footwear. Shoes protect against parasites, sharp objects, cold surfaces, and pathogens present in degraded urban soil. The risk calculus is not zero. It is an argument for understanding what was removed when the shoe went on, so that the removal is at minimum legible, so that when you feel the inexplicable ease that many people report when they step onto grass or earth in bare feet, you are not dismissing it as mood or nostalgia or the placebo of fresh air. The mechanoreceptors in your soul are activating an ancient neural circuit. The lamina I pathway is feeding signals into your interoceptive cortex. The terrain is demanding novel proprioceptive computation from your cerebellum. The soil aerosols are reaching your immune system. The cortisol axis is being modulated. The serotonergic brainstem neurons are receiving immune signals from organisms that evolved alongside the human microbiome. None of this requires belief. It requires contact. Go back to the moment you took your shoes off at the start of this. Whatever surface you were on, floor, carpet, ground, what you felt in that first second of contact was the Meissner's corpuscles and Merkel's discs firing as the texture registered for the first time without an intermediary layer. What you noticed afterward, a slight softening, a diffuse ease, may have been the autonomic signal beginning to shift. The body was always receiving this information. The shoe was blocking it. 5 minutes on Earth without shoes is not a minimum effective dose established by clinical trial. We do not have that trial. But it is 5 minutes of restored input to a system that was built over 3 million years to expect it. The brain makes very fast use of that signal. It always has. You just stop giving it the chance. Walk on soil tomorrow. Not because it is ancient. Not because it is natural. Because your brain is running a sensory architecture that was calibrated to that surface long before either of you existed. And it still knows exactly what to do when it feels it again.